Deoxyribonucleic acid, otherwise known as DNA, is held together with hydrogen bonds. A hydrogen bond is the intermolecular force occurring when a hydrogen atom that is bonded to a highly electronegative atom of one molecule is attracted to two unshared electrons of another molecule. It is composed of two long chains of repeating subunits called nucleotides. A nucleotide is a monomer of DNA. It consists of three parts, which are 5-carbon sugar, a phosphate group, and a nitrogenous base. 5-carbon sugar in a DNA nucleotide is called deoxyribose. The phosphate group consists of a phosphorus atom that is bonded to 4 oxygen atoms. The nitrogenous base contains nitrogen atoms and carbon atoms and is a base. How can only 4 nucleotides encode thousands of different genes? The sequence is almost like using the alphabet to spell different words. The 4 bases are thymine, cytosine, adenine, and guanine. In DNA cytosine, one strand pairs with the guanine on the opposite strand, and adenine pairs with thymine. These pairs of bases are called complementary base pairs. There are 2 reasons that complementary base pair is important in DNA structure. The first one is because hydrogen bonds between base pairs help hold the 2 strands of DNA molecules together. The second reason is because the complementary nature of DNA helps explain how DNA replicates before a cell divides. According to the base pairing rules, the order of nitrogenous bases on the nucleotides in one chain of the DNA molecule is complementary to the order of bases on the opposite chain. The order of nitrogenous on a chain of DNA is called base sequence. A replication fork is the Y-shaped region that results when 2 strands separate. As the replication fork moves along the original DNA, synthesis of one strand follows the movement of the replication fork. This then leaves gaps in the newly synthesized strand, which are later joined together by an enzyme called DNA ligase. Synthesis moves in opposite directions on complementary strands. DNA ligases are essential for the joining of Okazaki fragments during replication and for completing short-patch DNA synthesis occurring in DNA repair process. Okazaki fragments are short molecules of single-stranded DNA that are formed on the lagging strand during DNA replication. There are 3 steps to DNA replication. In step 1, enzymes separate the DNA strands. They move along the DNA molecule, breaking the hydrogen bonds between the complementary nitrogenous bases. The second step to DNA replication is DNA polymerase. DNA polymerase is when the enzyme makes a new strand of DNA that is complementary to the original. Semi-conservative replication is the final stage. In each new double helix, one strand is from the original molecule and one strand is new. It is called semi-conservative replication because each of the new DNA molecules has kept one of the original strands. RNA primers adds RNA primers onto the lagging strand, while Okazaki fragments are relatively short fragments of DNA synthesized on the lagging strand during DNA replication. A mutation is a change in the nucleotide sequence of a DNA molecule. When mistakes in DNA replication occur, that is when a mutation takes place. When mistakes in DNA replication occur, the basic sequence of the newly formed DNA differs from the sequence of the original DNA. There are several effects of mutations. The first one is that mutations can have a serious effect on the function of an important gene and disrupt an important cell function. Also, chemicals and ultraviolet radiation from the sun can damage DNA. The mutations that are not repaired can lead to several diseases, such as cancer. Cancer can occur if the mutations happen within genes that control cell growth and cell division. Mutations that affect genes that control how a cell divides can lead to an abnormal mass, otherwise known as a tumor.